Okay, so I'm going to go ahead and show you a couple little videos that show some of the biochemical pathways that we didn't really go over in the other. I'm not going to test you on these, but I think maybe it's good for you to see what's actually happening in case someday you are tested on it, so you'll at least have some exposure to the idea. So let's go ahead and get started. I'll speak as little as possible, which I know seems impossible with me, but I will try to keep that as much as possible. Okay, now notice real quick. So glucose actually had to enter into a cell. But remember, glycolysis, the first step, is happening outside of the mitochondria. So again, notice whenever we break a phosphate off and add it to something, we say we are phosphorylating something. And notice again, it took 2 ATP to start the process. But when we start splitting this into actually 2 different things and converting them over, notice we're actually starting to pull hydrogens out to form NADHs, and eventually we only get 2 NADHs from this process. We actually end up making 4 ATP, but we have to pay back those first 2, so we get a net gain of 2 ATP. And then, as long as there's oxygen available, then our pyruvic acid or pyruvate, our 2 3-carbon products, will enter the mitochondria. Okay, notice our 3-carbon product, pyruvic acid, loses a carbon dioxide, turns into our 2-carbon product, acetyl. Coenzyme A actually attaches to it, and when we're doing this, we are taking hydrogens out of this and converting NAD to NADH. So even at this step, the second step, we get some NADH and we pull carbon dioxide out of that to go from a 3-carbon to a 2-carbon. And now we're ready to go into the Krebs cycle. Okay, here we see our oxaloacetic acid, also called oxaloacetate, is the 4-carbon product. We're adding it, or the coenzyme is adding it to the 2-carbon product. And now we get our citric acid, also called citrate, which is our 6-carbon product. Notice 1, 2, 3, 4, 5, 6 carbons, which means these 4 add to those 2. Okay, we rearrange this. We come to our next product, which is still 6 carbons, isocitric acid. Okay, notice here to go from isocitric acid to alpha ketoglutaric acid, then notice we have 1, 2, 3, 4, 5 carbons from 6. As we removed a carbon dioxide from it, we also removed hydrogens from it to convert NAD to NADH. Now again, what's going on in here is the TCA, or also called the citric acid cycle, also called the Krebs cycle. Okay, again, we pull more hydrogens out. We also pull another carbon dioxide out, so I go from a 5-carbon to a 4-carbon product and I pulled carbon dioxide out and some hydrogens because I converted NAD to NADH to come up with succinyl coenzyme A. Okay, notice in this step we did make ATP, but it's really from converting GTP, pulling a phosphate off of it, which we're adding to ADP, so the GTP becomes GDP, the ADP becomes ATP. So all we really did was remove from this energy source to this energy source that phosphate we traded. Okay, and then we actually then have our succinic acid. Okay, we pull some more hydrogens out of here to go from FAD to FADH2, and we end up with fumaric acid. Okay, notice how we are adding water to the processes. Remember how we talked about both dehydration synthesis and hydrolysis reactions. So water is usually involved in most chemical processes in the body because we are mostly water, but we're converting fumaric acid to malic acid. And I'm finally pulling more hydrogens out of the malic acid, converting NAD to NADH, and then I'm back to my starting product, which is my oxaloacetic acid. Now again, notice that I've got 4 carbons here, so then the next acetyl that comes in from the pyruvic acid, I'm going to recycle this, and again we lost 2 carbon dioxides. The carbon from here or the carbon from here came from those carbons, and these 4 carbons kept being recycled through the process. Our last step, oxidative phosphorylation. Here's my mitochondria, the double membranes. I like this picture because it does show the inner membrane with our electron transport chain and our enzyme in it. It also shows the inner membrane space where our protons build up, and it shows our outer membrane, the outside membrane on the mitochondria. Therefore, when we build up the concentration of protons, they have to run back in. They can't just be escape into the cytoplasm. Okay, here we see our pumps pumping our protons into the inner membrane space. We see our electrons starting to move through the proteins. And again, when we reduce something, we've added a protein to it. When we oxidize something, we've removed a proton from it. Or excuse me, when we add a hydrogen to something, the same thing as adding electrons to it. When we remove the hydrogens from it, then we've oxidized it. Again, notice 1 pump, 2 pumps, 3 pumps. NAD gives up the hydrogen at the first area to power the first pump. The electron literally powers it and is passed from protein to protein to protein. The FAD doesn't start at the first pump. It starts at the carrier and powers the last 2 pumps. So again, I'm getting my pumps pumping the protons through here, the electrons moving through, powering the pumps. Then I literally have my oxygen come in, receive the electrons, also receive protons that are coming back in and being converted to water. Notice what was happening. This is my ATP synthase with the hydrogen ion channel built into it. The movement of the protons or hydrogen ions back through it energized this, which then allowed me to add phosphates back to the ADPs so I could form ATP. Again, another way of looking at the process with a little bit more detail and actual names for some of the proteins like cytochrome B, cytochrome C, cytochrome A, and my understanding is cytochrome C is one way we can actually tell relationships between different structures, because cytochrome C tends to be passed through the mother's DNA, and we can tell from species to species the exact order of the amino acids in that cytochrome C. So it really helps us develop relationships between things by understanding the development and the inheritance and the genetics of the cytochrome C. So that's just a little side note in case you ever hear about that. You'll know what they're talking about. Anyway, you don't have to know all the details here. I wanted you to see the details. You do need to understand the process that's happening, especially according to our earlier videos.